Hello friends, this is Dr. Kishore Kumar. Today I would like to present on the prokaryotic versus eukaryotic cells. In this particular topic, I am going to discuss about the cell theory, what do you mean by cell, and the cell theory which is very famous because that is relevant to discuss here, the cell theory concept. And at the end, I am going to discuss about the differences between prokaryotic and eukaryotic. In this particular session, I am not going to discuss about their structures, but the differences as well as the cell theory, which is I am going to be discussed. Clear? Then, first of all, you should think that what is cell. This is very important because we have to be understand what is cell. We know that cell is cell biology because this is what the life of a life or this particular organism is to be facing is having a life because of the cells. So what is cell? The cell is nothing but this is the simplest form of life. If we can define any particular organism, whether it is single cell or multicellular organism, that is to be manufactured with the cells. Or the cell is nothing but which is a symbol of life. Clear? Then this particular cell was first identified by the scientist Robert Hooke in his observation of the crop cells, where under microscopic, there at particular time there is a simple microscopes that can be possible and that can be present. So the simple microscopes scope was prepared by the Anthony van Leeuwenhoek. With the help of the simple microscope, this Robert Hooke first identified cell. So this particular cell, whatever we are calling, which is a form of life, is to be possible because of the after invention of light microscope. Because if you see the cell structure, that is that the cell size, which is a very minute, and we can't see with our naked eye most of the cells. We can't see with our naked eye because that are that much very small in size. So this cell biology has advanced when the discovery of this microscope has taken place. So the initial, which is Anthony van Leeuwenhoek, then later on there is a sophisticated research which is started in the field of microscope, and finally the electron microscope, and there will be a variety of different microscope which has come, so which are important for the understanding of the ultrastructures of the cell. So first we should understand that this cell theory, which is very important because the previous reports and the previous existing knowledge where the cells is limiting. So there is a famous theory that is proposed by the Sweden and Schwann. That name of our theory is cell theory, which is proposed in 1839. This cell theory, which is clearly state about three important points. So friends, this is very important. We should be understand. The first important point is all living things are composed of cells. That's what we discussed about. This is a form of life. So whenever you take any kind of the living cells, any kind of the living things, that should composed of cell or cells, maybe unicellular or multicellular. That is the first conclusion which is given by the cell theory. And second point is cells are the structural and functional units in living organisms. Whatever the functions or what are the structures that are with respect to the that living thing, all are into attributing to the cells. So whenever we call cell is to be a structural and functional unit of the life. That is the second point. And third important point, we should remember that the cells come from the other living cells. This is very important. So the cells will not come spontaneously or will not arise in spontaneously. So there is a clear-cut evidence later on which is proved that these cells, the existing cells, must have come from the pre-existing cells. Whatever the cells that are we are looking, these must have come from the pre-existing cells. So later on, this is identified by the number of experiments. The best example how the cell is arising from previous cell is cell division. That we should understand in the next previous classes. So all these three important points which are to be very important in order to understand the cells. Clear? So if you see this, the whole cells, once the cell theory has been formulated and the definition of cell, once we come to know, then there is a question that how many different type of cells that can be present in the nature. So this is very important. If you take all living cells that are present in the nature, that can be divided into non-cellular and cellular part. So which is majorly division into non-cellular to cellular. So if you take the non-cellular for the best example is virus. So-called we call viruses, various different kinds of viruses. These are all comes under the non-cellular cells. So non-cellular mean we generally don't define the virus is a cell, right? So that's what non-cellular cells, the best example is virus. If you take the cellular level, which is again for an easy understand, it can be divided into two different types. First one is to be prokaryotic cell and second one is to be eukaryotic cell. So what do you mean by prokaryotic cell? Prokaryotic cell means this is to be a very primitive organisms and that are manufactured with the single cells, right? That we call as single cell microorganisms. So single cell microorganisms are comes under the prokaryotic cell. This best example which we are discussing is bacteria, right? You take the bacteria is having a life that is manufactured with the only single cell. So unicellular organisms are prokaryotic cells, right? Their structures and everything we will see into the next class. And second one, if you see the eukaryotic cells depending on their structures, it can be further classified. Among them, first one is fungal cells. These are to be the best example of eukaryotic cells. Then next one is to be animal cells, like humans also, and the other type of the eukaryotic cell is to be plant cells. So you should remember this is a very basic introduction. You just have an idea about what different kinds of the cells that can be present in the nature, right? So eukaryotic cells are fungal cells, animal cells, and plant cells. Whereas non-cellular, the best example is viruses. Clear? Now, we will see what are the different types of the differences which can be present between the eukaryotic and prokaryotic cells. Because you can see in many entrance exams, even from the 7th to 8th standard to the at maximum PhD entrance levels also, the n number of questions which is being asked from this, the differences between prokaryotic and eukaryotic. So it's better to remember the basic points in what point of view these two cells are different. So coming to the definition, any cell that contains clearly defined nucleus and membrane bound organelles, these are called eukaryotic cells. Clear? So whereas prokaryotic cells, any unicellular organism that does not contain the membrane bound nucleus are organelles. This is very important. So if you take eukaryotes, as I explained previous slide, if you take plant cell or animal cell, even human cell, where there is a very clearly defined cell nucleus that can be present. Where exactly the nuclear material that can present in the nucleus. Clear? Apart from that, there will be n number of cell organelles that can be present in eukaryotics, but in a prokaryotics which are not present. Clear? Anyway, this we can better understand when we go into the structures of prokaryotic and eukaryotic cells to the coming classes. And example is, as I explained, animal cell, plant cell, fungi cell, protista cells. And whereas in a prokaryotic, bacteria and archaea, these are the unicellular organisms that are best examples of prokaryotes. If you take the nucleus, definitely the nucleus that can be present because as we seen in the definition, that is to be membrane bound. Membrane bound means which is a double membrane that can be manufactured, and inside is nucleic acid is present. But whereas the nucleus that is absent in prokaryotes. And if you see the size of these cells in eukaryotes, these are larger size. That can be around 10 to 100 micrometers. But whereas prokaryotic, the small size that can be typical to the prokaryotic, and if you see the measurement, that is below than 5 micrometers. And come to the DNA replication, which is a highly regulated with the selective origins and sequences that can be present in eukaryotic cells, whereas prokaryotic cells replicates entire genome at once. Clear? Because in prokaryotes, the genome is not to be a very typical length. So that is to be very small in length. In this particular case, what is happening, the entire replication will complete at the same time. But in eukaryotes, that is not possible because various number of origins that can be present, multiple origins can be present, and it is trying to initiate at different locations at the multiple times. And this can we understand at DNA replications. And organism type, obviously eukaryotic are multicellular, means more than cell that can be present. If you take humans, human body is composing of trillions of cells, 100 trillions of cells that can be present. But prokaryotic, these are a unicellular, only single cell that can be present in these organisms. And if you take the chromosomes, definitely more than one chromosome, which is a typical feature of eukaryotic cell. But whereas prokaryotic cell, one long single loop DNA, that is nothing but a single chromosome that can be present. Along with this, there is an extracellular, extranuclear material that can also be present. This we call plasmids, right? The plasmids are exclusively present in prokaryotes, but these are absent in eukaryotes. So these plasmids are very important for the technology of genetic engineering that we will discuss later. Right, come to the ribosomes. These are protein manufacturing factories that are very large size in eukaryotes and very small size in prokaryotes. If you see in eukaryotes, that is 60 ribosomes that can be present. But anyway, the structure which I am going to discuss in detail coming classes. Right, the growth rate and generation time, obviously eukaryotic cell is to be very less because which is to be a complex structures, and whereas in the prokaryotes, this is a very faster time. If you take E. coli, only that will take 20 minutes for division. So one E. coli bacteria within 20 minutes, it is divided into 2 E. coli bacteria, right? So that's what which is very faster. And if you see the organelles, as I discussed, organelles are present in eukaryotes and these are absent in prokaryotes. So you can think that organelles mean what different kind of organelles? Endoplasmic reticulum, Golgi complexes, and even chloroplast, even plasts. There will be n number of lysosomes, n number of organelles that can be present in eukaryotes, and these organelles are absent in prokaryotes. And ability to store hereditary information, obviously eukaryotic cell can store it, and even prokaryotes also they can store. That is during cell division, it can be passed on to its daughter cells. And coming to the cell wall, in eukaryotes, if you take animals and humans, they don't have any kind of cell wall. But the cell wall is present for the plant cells, that we should remember. plant and fungi cells will have the cell wall, but animal and human cells, they don't have the cell wall. But whereas the prokaryotic cell, this is the cell wall is to be complex, and this can compulsory present in the prokaryotic organisms. And if you take the plasma membrane, obviously that can be present in eukaryotes as well as prokaryotes. If you see the lipid bilayer structure of this plasma membrane, which is to be more or less same in prokaryotic as well as eukaryotics. And if you see the cytoplasm, definitely that can present in both the organisms, that eukaryotic and prokaryotics, right? So in this particular video, I'm trying to explain about the very basic concepts of prokaryotic and eukaryotes and the cells and their differences. So students, I'll again meet you in the next class. Then see you. Bye.